Bismillahir Rahmanir Rahim. Dear students, Assalamu alaikum. Last time we discussed two experiments. One was the lab safety measures, and the other was the microscope, the parts of the microscope, and the functions of these parts. Today we are going to make a slide. Today I will tell you, and when you will come in the university, inshallah ta'ala, in the lab, you will perform that experiment. So today we are discussing what we are discussing today. Today we are going to make a specimen, a specimen, a specimen. What is a specimen? Aina. A specimen also called mount. A specimen also called mount. So this specimen is of two types. One is daimi, permanent. Yani we make them we make the specimen one time and we can use the specimen for long time. Second type of specimen is called temporary specimen. What is temporary specimen? Temporary specimen is we make the specimen, and when we finish using, either we throw it or we wash it and we can use it again. So today what we are discussing today is actually temporary mount. Mount yani specimen, same thing, slide. These slides, they are of two types. Two types. One type of slides we make using a drop of water. These slides are called wet mount or wet specimen. Wet mount. Wet mount. This is wet mount. Okay. So this is one type of slide. The other type of slide is we make without using water. Dry. Nashif. Okay. So specimen two types. يعني أين بالموية أو أين بدون موية يعني ناشف. So today in this experiment we are discussing wet slide. The next experiment will be about dry slide. Okay, so today we are discussing the wet mount. What is wet mount? I will discuss. Dear students, first of all, the thing we want to discuss is resolving power. Resolving power. Resolving power is also called resolution. So all of us have mobile phone. In mobile phone we have camera. Camera has got resolution. Masalan, this is my camera. Resolution is 5 megapixel. This is your camera, iPhone. Resolution, masalan, I'm just telling you example. This is your camera, 20 megapixel. So you can see which image is more clear. I think this image is more clear. You agree? It means that this image has got more resolution power. Okay, so resolving power is actually the ability of the microscope, ability of the machine to see the object clearly. Now the resolving power of human eye, our eye, is 0.5 millimeter. Yani we can see up to 0.5 millimeter. If the distance between any two objects 0.25 millimeter, we can see them separately. Otherwise, if it is less than 0.25, we will see it as one object. Okay, this is called resolution power. The microscope resolution power is 0.25 micrometer, so small. It can see smaller objects. And the electron microscope 0.5 nanometer. So it means that it can see more small objects. Okay, so it means that the microscope, both the light microscope and electron microscope, will help us to see very small objects, the object that we cannot see through our eyes. Second thing is refractive index. You know refraction? It is inkisar, mu'alaja inkisar. Okay, inkisar or refraction is the ratio of speed of light in the air as compared to any other medium. Okay, so this is very important thing to understand. What is a refractive index? Wet mount yani ayna bil moya, we usually use to make slides when we are dealing with any liquid. For example, saliva, blood, urine. Saliva yani loab, blood yani dam, urine yani bol. So if we make these type of slides, we make wet mount. Wet mount yani ayna bil moya. In this experiment, look at the ihtiyajat. You look at the requirements: microscope, glass slide, cover slip, scissor, qus, scissor, needle, and qatara, dropper. These are the objects that we need to perform our experiment. For today experiment is wet mount experiment. Wet mount experiment, what we need a slide, glass slide. We need cover slip. So you can see cover slip also, and you can see distill water, moya. Okay, because this aina bil moya. Okay, so how? First of all, you take the slide, then you put a specimen upon the slide. Here you can see this specimen is actually a paper, waraka, sagir. Okay, but this is just to show you the procedure. In the next experiment, in place of this paper, you will use the cell. So today experiment is just to show you the procedure. That is why we are using this paper. Or you can cut any one letter. For example, letter E. If you cut, you can make the slide. But this slide is not real. The real slide we will make in the next experiment. Okay, sir? So this is, he put the slide. For example, this is Aina. And now he will put 1 or 2 drops of water. 1 or 2 drops of water he will put. Okay, and then he will put cover slip. How to put the cover slip? See the procedure of putting the cover slip. How he will put the cover slip? You will put the cover slip at an angle of 45 degrees, 45 daraja, and then slowly put it down. Okay, so first you put ala daraja 45 and then nazal shoya shoya. If you don't do it like this, there will be air bubble in the slide and this slide will not be good. So what you see on the screen, this is a good slide because there is no air bubble here. Now you will see the slide in the microscope. You will put this slide in the microscope. Always start from 4x, the smallest. And you know 4x is called scanning lens. Scanning yani scan. When you use 4x, it is for scanning. Final aina. Final makan aina. So you locate the aina, locate the specimen. It is the function of 4x. Takbir will start, magnification will start from 10, also called low power, and then 40, also called medium power, and then 100, also called high power. So we will just locate the aina using 4x, and then we will move to 10x. When you move to 10x, see now he is focusing the slide. He is focusing the slide, okay, by moving the coarse focusing knob. And now this letter E is focus. Okay, this is the letter E that you see in the microscope. Okay, in low power yani 10x. Now when you will increase quwwat takbir to 10x, you will increase. Also you can change the light by using the diaphragm. Okay, now for next lens, next quwwat takbir, if you see, what do you see in the microscope now? What do you see in the microscope? This is the object you see in the microscope. So what is the difference between the last yani picture and this picture? What is the difference between last picture and this picture? Last picture, letter E, you saw complete, kamil. But when you increase Quwwat Takbir, when you increase Quwwat Takbir, because of the zoom, you can see only juzv, haga surah. Only the part of the picture, not the whole picture. This is called field of view. So it means that Quwwat Takbir and field of view, Medan al Roya, bil-aks mutanasib. Bil-aks mutanasib. Inversely proportional. When you increase Quwwat Takbir, field of view decreases. Now, now if you increase the power more, if you increase the power more, what will happen? When you increase the power more, what will happen? Now see, this is letter E. Only you can see the small part of letter E. It means that when you increase Quwwat Takbir, when you increase Quwwat Takbir, field of view, yani the part of the picture that you see decreases. Okay. Now see, he is now showing you the field of view. In a smaller field, you can see the complete picture of this person. But if you increase the Quwwat Takbir, now you can see only a part of his body. You cannot see the complete picture. This is called field of view. So it means that when you increase Quwwat Takbir, field of view decreases. I think you understand this thing. Okay. So dear students, I just show you the method of making wet mount. Okay, this is the same method shown here. This is the same method shown here. Yani, you will put one drop of water upon the slide, then put the specimen. In this case, it was letter E. Then you put the cover slip at an angle of 45 degrees and decrease the cover slip slowly. This will complete the wet mount slide, and then you will put the slide in the microscope and start from 4x, locate where is the letter E, and then you increase the power to 10 to see the picture of letter E. When you will increase it more, the picture will be zoomed and you can see only a part of it. But dear students, can you notice something in this picture? This picture is showing letter E, but this letter E is opposite, not straight. You know why? Because of this property of the microscope, refractive property. Refractive, what is refractive property? The image which is formed is opposite. Opposite. It means that when you will move the slide right or left, what you see in the microscope, it will be opposite. Yani if you move the slide right, what you see in the microscope, the aina will move to the left. If you move the slide forward, then what you see in the microscope, aina will move backward. This is called refractive property. This picture is shown to show the same thing I just told you. This is less power, more power, more power. So the difference is the field of view. When you increase field of view, when you increase Quwwat Takbir, field of view decreases. So dear students, this completes my first experiment. If you have any question, you can ask me. What is the method of the experiment? I am just showing you one movie